Welcome, everyone. I'm excited to kick off today's presentation on Respiration. This is the first slide, so I want to warmly welcome you all and thank you for being here. The title of our talk is simply Respiration, and we'll also explore some related concepts along the way. In this session, we'll cover the basics of respiration, why it's important, and how it impacts our health and daily life. But don't worry, I'll keep it engaging and straightforward. Let's get started. Let's begin by looking at the different types of respiration. We have aerobic respiration, which is the most common and efficient form, and anaerobic respiration, which occurs in environments lacking oxygen. Moving on, we'll explore cyanide-resistant respiration, a special pathway some organisms use to survive in toxic conditions. Next, we'll delve into the formation of acetyl-CoA, a crucial step that links glycolysis and the Krebs cycle, serving as a gateway for energy production. Understanding the steps of aerobic respiration is key, as they include glycolysis, the Krebs cycle, and the electron transport system. The electron transport system is where most ATP is generated, harnessing energy from electrons transferred through a series of proteins. The Krebs cycle, also known as the citric acid cycle, is vital because it breaks down acetyl-CoA to produce energy-rich molecules. We'll also discuss the significance of respiration itself, how it powers cellular activities and sustains life. Lastly, the respiratory quotient gives us insight into the type of substrate being metabolized based on the ratio of carbon dioxide produced to oxygen consumed. All these points connect to give us a comprehensive understanding of how organisms convert nutrients into usable energy efficiently and adaptively. Let's take a closer look at the regions involved in respiration in plants. Here, we see floral and vegetative buds, germinating seeds, as well as stem and root apices. These are all actively respiring regions, meaning they're engaged in the process of breaking down stored nutrients to generate energy. Speaking of nutrients, the substrates that plants use for respiration include carbohydrates, fats, and proteins. These are the main types of organic molecules that provide the energy necessary for growth and development. It's important to understand that respiration requires a substrate, so it depends on the availability of these nutrients. Under normal conditions, carbohydrates like sugars are the primary substrate, but fats and proteins can also be used, especially during stress or specific developmental stages. Now, the overall chemical reaction for respiration can be summarized as glucose combining with oxygen to produce carbon dioxide, water, and a significant amount of energy, 686 kilocalories per mole. The basic equation is C6H12O6 plus 6O2 yields 6CO2 plus 6H2O, and the release of energy. This process essentially breaks down complex organic substances, like carbohydrates, fats, and proteins, to release CO2 and water, making energy available for various cellular activities. So, in summary, respiration is vital because it provides the energy necessary for plant growth, maintenance, and development, especially in those actively growing regions we just discussed. Let's take a closer look at anaerobic respiration. This process kicks in when oxygen is scarce or unavailable, and cells still need to produce energy. It's one of the key types of respiration, alongside aerobic respiration. The main idea here is that anaerobic respiration allows organisms to generate ATP, which is their energy currency, without relying on oxygen. It's especially important for certain microorganisms and in situations where oxygen levels are low, like in some muscles during intense exercise. In essence, anaerobic respiration can be seen as a backup system that keeps cells functioning when aerobic respiration isn't an option. This ties into our overall theme of how different organisms adapt to their environments to meet their energy needs. So, understanding these different types of respiration helps us appreciate the versatility and resilience of life at the cellular level. Let's take a closer look at anaerobic respiration. This process takes place exclusively in the cytoplasm, meaning it doesn't involve the mitochondria. When one molecule of glucose undergoes partial oxidation through this process, it only produces about 2 ATP molecules, which is quite a bit less than what we see in aerobic respiration. The end products are mainly carbon dioxide and either ethyl alcohol or lactic acid, depending on the organism. Interestingly, in this process, glucose is only partially oxidized, which explains the lower ATP yield. You'll find this type of respiration in some bacteria, fungi, and certain endoparasites. A key point here is that oxygen is not utilized at all during anaerobic respiration. This is a crucial distinction because it allows these organisms to survive and produce energy in environments where oxygen is scarce or absent. Understanding the differences between anaerobic and aerobic respiration helps us appreciate how diverse life forms adapt to their environments and optimize energy production under different conditions. Fermentation, as we see here, is actually a form of anaerobic respiration carried out by microorganisms. It's a fascinating process because it allows these tiny organisms to produce energy even in the absence of oxygen. Examples of fermentation include alcohol fermentation, lactic acid fermentation, and acetic acid fermentation. What's interesting is that during fermentation, stored food is oxidized into specific compounds rather than producing carbon dioxide and water like in aerobic respiration. For instance, in lactic acid fermentation, glucose is converted into lactic acid and a small amount of energy, specifically 2 ATP molecules. Similarly, in alcoholic fermentation, glucose is transformed into ethanol, carbon dioxide, and energy. This process is crucial in brewing and baking industries. The key point here is that anaerobic respiration involves the partial oxidation of glucose without oxygen. Instead of fully breaking down glucose into carbon dioxide and water, the process stops midway, producing different compounds like lactic acid or ethanol, along with some energy. So, in essence, anaerobic respiration is a vital alternative pathway for energy production in situations where oxygen is scarce or absent, connecting directly to the larger theme of how organisms adapt their metabolism to different environments. Aerobic respiration is a process where molecular oxygen is used to completely oxidize glucose. This means that the food stored in our bodies is fully broken down into carbon dioxide and water. In doing so, a significant amount of energy is released, which our cells harness to perform various functions. The overall chemical reaction can be summarized as glucose reacting with oxygen to produce carbon dioxide, water, and a substantial amount of energy, specifically around 686 kilocalories. This process is essential because it provides the majority of the energy our bodies need for everyday activities, growth, and maintenance. It's a highly efficient way of extracting energy from the food we consume, and it's a key part of understanding how our bodies turn food into usable power. Now, let's look at the respiratory quotient, or RQ, of some common organic compounds. As you can see here, tartaric acid has an RQ of 1.6, which indicates it produces more carbon dioxide relative to oxygen absorbed during respiration. Oxalic acid and citric acid both have an RQ of 1.33, suggesting a similar pattern in their metabolic processes. Malic acid has a lower RQ of 0.7, meaning it consumes more oxygen compared to the carbon dioxide produced. Fats have an RQ of 0.7 as well, which reflects their high oxygen requirement during oxidation. Carbohydrates, on the other hand, have an RQ of 1, showing a balanced ratio of CO2 produced to O2 absorbed. The formula for RQ is really straightforward. It's the volume of carbon dioxide evolved divided by the volume of oxygen absorbed. Understanding these values helps us grasp how different compounds are metabolized and how their oxidation processes vary, which ties into the overall theme of energy production and metabolic efficiency in our bodies. All right, now let's take a closer look at the steps of aerobic respiration. As you can see on the slide, it consists of 3 main stages. First, we have glycolysis, which occurs in the cytoplasm and breaks down glucose into pyruvate. This process produces a small amount of energy in the form of ATP and NADH. Next, the pyruvate enters the mitochondria and is further processed in the Krebs cycle, also known as the citric acid cycle. Here, more energy carriers are generated, and carbon dioxide is released as a waste product. Finally, the electrons from these carriers are transferred through the electron transport system, or ETS. This is where the majority of ATP is produced, using the energy from electrons to drive the formation of ATP through oxidative phosphorylation. These three steps work together seamlessly, enabling the cell to efficiently extract energy from glucose during aerobic respiration. Glycolysis, also known as the Embden-Mayer-Hoff-Parnas pathway, is a fundamental process in cellular metabolism. It's often referred to as the common respiratory pathway because it's shared by both aerobic and anaerobic respiration. Essentially, this pathway involves the breakdown of glucose, which has 6 carbon atoms, into 2 molecules of pyruvic acid, each with 3 carbons. One of the key points to remember is that this process occurs in the cytoplasm of the cell, making it accessible and efficient. Importantly, glycolysis does not require oxygen, which means it can proceed under both oxygen-rich and oxygen-deprived conditions. This versatility highlights its vital role in providing energy swiftly, regardless of the cell's environment. Connecting this to the overall theme of respiration, glycolysis is the first step in extracting energy from glucose, setting the stage for further pathways to generate more ATP and energy carriers. Now, let's take a look at what happens when pyruvic acid breaks down. As you can see, pyruvic acid can be converted into several different compounds depending on the conditions in the cell. For example, it can become alanine, which is an amino acid important for protein synthesis. It can also be transformed into ethanol in some organisms, especially during fermentation. Another key pathway involves amino acid synthesis, where pyruvic acid serves as a building block. Additionally, pyruvic acid enters the Krebs cycle, which is central to energy production in our cells. During this process, pyruvate is further broken down, releasing carbon dioxide and water, and generating ATP, the energy currency our bodies rely on. In anaerobic conditions, pyruvic acid can be reduced to lactate, which allows glycolysis to continue and produce energy when oxygen is scarce. Overall, the breakdown of pyruvic acid is crucial because it links several metabolic pathways, helping our bodies adapt to different energy demands and environmental conditions. Now let's talk about the formation of acetyl-CoA, which is a crucial step in metabolism. As you can see, acetyl-CoA is a 2-carbon compound, and it acts as a key link between the Embden-Mayer-Hoff pathway, or EMP, and the Krebs cycle. Essentially, after glycolysis produces pyruvate, this pyruvate is converted into acetyl-CoA. This conversion is vital because it feeds the Krebs cycle, where further energy production occurs. So, in a way, acetyl-CoA is the bridge that connects the breakdown of glucose to the energy-generating processes in the mitochondria. Understanding this step helps us see how different metabolic pathways are interconnected, ensuring efficient energy extraction from nutrients. The Krebs cycle, also known as the TCA cycle, organic acid cycle, mitochondrial respiration, oxidation of pyruvate or citric acid cycle, was discovered by Sir Hans Krebs. This cycle is fundamental to how our cells generate energy. In this process, acetyl-CoA reacts with oxaloacetate, which has 4 carbons, to produce citrate, which has 6 carbons, and this is the first stable product of the cycle. The entire process takes place in the matrix of the mitochondria, which are often called the powerhouses of the cell. An important point to remember is that oxygen is involved in the cycle, making it a key part of aerobic respiration. So, in essence, the Krebs cycle is a crucial step in converting nutrients into usable energy, linking the breakdown of carbohydrates, fats, and proteins to the production of ATP, our cell's energy currency. Now, NADH and FADH2, which are produced during glycolysis and the Krebs cycle, are actually oxidized back to NAD and FAD. During this oxidation process, electrons are transferred through a series of electron carriers, such as coenzyme Q and the cytochromes B, C, and A3. As these electrons flow through the chain, they drive the phosphorylation of ADP to ATP by adding a phosphate group, pi. This process is what we call oxidative phosphorylation, because it couples the oxidation of NADH and FADH2 with ATP synthesis. The electron transport system, which is the third step of respiration, takes place in the inner mitochondrial membrane, specifically within the cristae. This location is crucial because it provides the surface area necessary for efficient electron transfer and ATP production. Understanding this step highlights how tightly linked electron transport and ATP synthesis are, and it really underscores the importance of the mitochondria as the powerhouses of the cell. Now, let's look at the significance of respiration. First, respiration releases CO2, which plays a crucial role in maintaining the balance of the carbon cycle. This process helps ensure that carbon is recycled efficiently in nature. Next, respiration also contributes to the formation of other essential compounds, acting as an important part of the cell's structure and function. It's vital for creating the building blocks that cells need to operate. Additionally, respiration converts insoluble food into a soluble form, making nutrients more accessible for the plant's use. And finally, it releases energy that is used in various metabolic processes. This energy is essential for the plant's growth, development, and overall functioning. Altogether, respiration is a fundamental process that supports plant life and sustains the balance of ecosystems. Let's discuss the role of cyanide-resistant respiration in plant processes. It's interesting to note that this pathway is essential for peroxide-catalyzed reactions involved in ethylene biosynthesis, which plays a key role in fruit ripening. This means that cyanide-resistant respiration is not just a backup pathway, but actively contributes to the ripening process. Furthermore, it's believed that this alternative respiration helps keep the Krebs cycle running smoothly. It does so indirectly by supporting the oxidation of NADH and the regeneration of NAD, which are crucial for energy production and metabolic continuity during ripening. Now, when electrons are transferred through the electron transport chain, they pass from ubiquinone to flavoproteins, then to an alternate oxidase, and finally to oxygen. Interestingly, instead of producing water, this process may generate hydrogen peroxide, which is thought to be responsible for the climacteric phase in fruits, essentially a burst of ripening activity. Hydrogen peroxide, in turn, accelerates membrane oxidation and breakdown, facilitating the ripening process. In higher plants, cyanide inhibits cytochrome oxidase activity, but in such cases, this alternative oxidase pathway becomes active. It essentially bypasses the inhibited complex and provides a different route in the electron transport chain, possibly diverging from ubiquinone. Overall, cyanide-resistant respiration is a vital adaptive process that supports fruit ripening by maintaining metabolic flow and generating signals like hydrogen peroxide. It highlights how plants can switch pathways to ensure continued energy production and developmental processes even under stress conditions.